How's everyone doing today? Great, awesome. Happy homecoming. Good afternoon, everyone. Thank you so much for being here. My name is Sahil Singh, and I am a Master's of Engineering in Mechanical Engineering. This is my first year here, and if you know anything about the MEng program, it is also my last year here, so I am making the most of it. But I want to share a little bit with you about why I love Berkeley so much. And I did an undergrad in Florida in a different institution, and I did it in aerospace engineering. Aerospace engineering is a field where you have to think outside the box. You have to think outside the world, and you have to look for innovative solutions because we're writing the next chapter of space exploration right now. However, I found that in undergrad, I wasn't able to explore my interests as much as I could. And that all changed when I came here to Berkeley. My sense of curiosity when I arrived at this very special place went through the roof. There are so many incredible people here, so many incredible perspectives to embrace, and you're about to hear one of them today. Without further ado, I'd like to introduce our speaker for today, Professor Sarah Stanley. Here's a little bit about her. Professor Stanley is an associate professor at UC Berkeley with joint appointments in the molecular and cell biology and the School of Public Health. Her research focuses on how the immune system responds to Mycobacterium, tuberculosis, also known as MTB, which is the leading cause of infectious death worldwide. Her lab investigates how MTB evades immunity, what constitutes protective responses, and how this knowledge can inform new vaccines and therapies. Dr. Stanley also serves as the faculty director of the Alliance for Global Health and Science, which builds scientific capacity in East Africa through research and teaching opportunities. Without further ado, please welcome Professor Stanley.
Thank you so much for that introduction. And it's really great to be able to be here today. I always like to start a talk by getting a feel for who's in the audience. So how many of you here today are Berkeley alum? Awesome, welcome back. How many of you are biology majors? How many of you are current students? Oh wow, a lot of current students. How many of you have taken a class with me? Oh, okay, good. No, well, maybe you will after today. So I'm going to tell you today about the disease that I work on, which is tuberculosis. I'm going to tell you a little bit about the history of the disease, give you a sense for what is happening today in the world, and then I'm going to talk a little bit about research. The research that's going on that graduate students and postdoctoral fellows in my lab are conducting right now. So this is a little bit of a technical talk. There will be some data in this talk, and what I really encourage is for people to ask questions. So at any point in the talk, please interrupt me if you have a question. When I teach my classes, I love when people raise their hands and ask questions. Even if I don't get through all my slides, my goal for today is for you to learn something about tuberculosis and for you to appreciate that this is a really important problem. Okay. I'm also going to ask some questions, but I promise I won't call on anyone. So I want to start by asking you to take yourself in your mind back 100 or a couple hundred years. Imagine that you're in Victorian England. Okay, so this is a picture of a slum in London in 1830. What was going on at this time was the rise of industrialization. Cities were growing exponentially. They were becoming more crowded. And you saw the appearance of these things that at the time were called slums, where lots and lots of people would crowd into housing, right? A whole family might sleep together in one room. There was no ventilation or not adequate ventilation. There wasn't sewage. And so infectious disease was really rampant at this time. So anyone want to take a guess at what were some of the major infections that you might have been exposed to if you lived in London in 1830? Smallpox, great, good. Any other ideas? Cholera, oh good, yeah, okay. And? Malaria, maybe not as much. Many parts of the world, it was and is a huge problem. What else? Typhoid fever. Well, this is a very sophisticated audience. This is true, all of these diseases. The reason I wanted to start this talk today is to just remind people what it was like before we had cures, right? A lot of people died of infectious disease. And the average lifespan back then was not what it is today. I think today, this is a little bit of an aside, but I think today we've kind of forgotten how dangerous infectious disease can be. Okay, so I'm going to tell you today about an infectious disease that was a huge problem at the time. So probably most of you have heard of tuberculosis. Many of you may think of it as a disease of the past. A lot of people in the US think of tuberculosis as a disease of the past, and many of us know that it was once a really huge problem in Europe, in the United States, and other parts of the world. So this is actually a cartoon from Ireland depicting tuberculosis here as the Grim Reaper. And then surrounded, I think this is a little hard to see, but surrounded by these wolves that had what were thought to be the causes of tuberculosis. And so you can't read this text, but it says unsanitary workshops, slum tenements, tuberculosis milk. I have no idea what that is. Maybe unpasteurized milk, right? And the reason that the march of the White Plague is because it's estimated that 1 in every 4 people died of TB during the 19th century. And so TB was something that really permeated everything. How many of you read the book Everything is Tuberculosis? Yeah, that's a great book, right? I think it probably gave you a sense for how much tuberculosis has shaped history and culture. And actually at the time, so one of the things about tuberculosis that actually is very tragic is that it tends to strike people when they are young adults at the prime of life, at the peak of creativity. Although saying this as not a young adult now, I don't want to say I'm past my peak of creativity. But what would happen is people would get sick, and then they would have this, they would know they had tuberculosis. They knew it was a death sentence, right? Like many of you have probably seen a movie, or maybe you've seen La Boheme, right? Where the heroine or the hero coughs into a handkerchief, and you see the blood, and that person knows that, their time is, they don't have much time. And so it was very romanticized. It was associated with, with these bursts of creativity and passion. And so it was just part of the culture. So back then, people didn't understand how TB was transmitted or even what it was, okay? And today, what we know is that tuberculosis is an infection primarily of the lungs. It's caused by a bacterium. So when a person who's infected with TB gets active disease, and I'll tell you in a minute what I mean by active disease, they cough out aerosols. Those aerosols, if they're breathed in by someone who doesn't have TB, that bacteria goes into their lungs and then can cause infection. And it's estimated that one person that's infected with TB will infect 20 other people. So we know that the phenomenon, which we used to call slums, and now we, in academic settings, at least we now refer to as informal housing, this still happens in the world today, right? There are lots of parts in the world, in lower income countries, where there is still housing where many, many people crowd into small spaces. And access to medical care, access to the things that we enjoy as privileges in the United States are not present. And so for that reason, TB is still a leading cause of death globally. So it's estimated that there are about 10 million new active cases of TB every year. So what this means is that people actually have symptoms of disease. They're sick, they're coughing, they're transmitting the bacteria. And there are about 1.5 million deaths every year from TB. So part of the reason, okay, let me just ask, how many of you were aware of that? Of the toll that TB takes globally? Yeah, not very many people, right? Not even half the people in this room raise their hand. And one of the reasons for this is where this disease occurs. So this is a map that's looking at the number of cases that happen every year of tuberculosis. And so what you can see is that the largest number of cases occur in South Asia, Southeast Asia, and China. But the highest burden of cases, if you normalize it to the percent of the population, is in sub-Saharan Africa. Okay? So that's where a lot of the TB disease still happens. I actually want to take a minute and just take a minute to spend some time on that point. So TB kills 1.5, 1.7, something like that, million people every year. COVID-19 killed a lot of people, right? But not that many more than that, even in the worst year of COVID. So the whole world mobilized for COVID-19. Every vaccine strategy that exists was tried. Every company in the world was looking for drugs. But we have this disease, tuberculosis, that's killing that many people every year, year after year, and yet it's very neglected. Okay, so, but why don't we have TB in the United States anymore? So this is looking at the top 10 causes of death in 1900. Okay, so here you can see as a lot of the things that are causing top 10 causes of death in the United States in 1910 or 1900 are pneumonia, gastrointestinal infections, and tuberculosis. So tuberculosis itself was a major contributor. But now, if we look at 2010, there's almost nothing here that's a communicable disease, an infectious disease. And we have the things that we're all well aware of in this room. Cancer and heart disease are major causes of death. So we've managed to almost eradicate TB from the United States. Okay, any guesses how or why? Vaccination, I wish that were the answer. We do not have a vaccine for TB, yeah? Sanitation is important, so part of it may be that we live in big houses that are well ventilated and we're not, so it spreads a little less easily. Public health measures are very important. I'm going to talk about that on the next slide. Any other thoughts? Antibiotics, good. Okay, so we're going to talk a little bit about all of these things. Okay, so if you get TB in the United States, what happens? If you're coughing, you go into your doctor. The doctor's able to figure out that you have TB. You're coughing, you have night sweats, you have fever, you might have weight loss. You get a chest x-ray, you get diagnosed with TB. What then happens is that there's a very robust public health response that's triggered. So you may be isolated. You may be told that you have to go home and isolate. Then what happens is that public health officials will say, who do you hang out with, right? And they'll go interview all those people and figure out if any of those people need to be tested for TB. And then if those people have TB, they will get isolated, right? So the local and state health departments are really important for this, and this is one of the ways that we've been able to keep TB cases low. So one of the things that we know is that when these systems fail in the United States, we can have outbreaks of TB here. So how many of you are aware that there was an outbreak in Kansas last year, a TB outbreak in Kansas? Yeah. So this is a little bit outdated, but this is actually the only headline I could find. So there were somewhere around over 100 cases of TB that were detected in Kansas. And that is, in the news they were saying this is the biggest outbreak that's ever happened in the United States. No, most people used to have TB in the United States. But in the modern times, right, since we started, we have all the tools that we have, this is a really big outbreak. So maybe it doesn't seem like that scary, but to people who are experts in the field, this is really alarming. Because what it says is there's been some breakdown in our systems. Yeah? Is mortality still super high in those cases? I think that a couple people died in that outbreak. And one of the things about TB is that if you have drug-sensitive TB, and I'm going to talk a little bit more about that, and you get treated in time, the chances that you'll survive the infection are pretty good. I'm going to tell you in the second half of my talk today that there's a lot of drug resistance. And so we're very lucky that this wasn't a drug-resistant outbreak. Yeah?
How does it start with like patient zero?
Where does it come from? Often in the US, it comes because someone comes to the US from a country where TB is endemic. Not always, but often. Yeah?
But sometimes some people will have it, right? It will it will trigger when they have this in whatever environment. When they have it, like.
Latent. Are you talking about they have it, but they, yeah? Okay, good. Yes, I'm going to tell you about the disease in just a moment. But these are the challenges to eradicating TB. So TB in many ways is a disease of poverty. If we could develop the world to the same standards that we enjoy in the United States, we could get rid of TB. That's a really hard problem, right? There's no vaccine. So some of you may have heard of a vaccine that's called BCG. Maybe some of us in this room have been vaccinated with BCG. It's still widely used in TB endemic countries. It's widely used because it can protect children from severe forms of disease, but it doesn't protect you into adulthood. So we don't have an effective vaccines. HIV and TB have a very deadly synergy. If you have HIV and TB, your TB is extremely severe. The drugs that we have are very challenging. So if you have drug-sensitive TB, you have to take this many pills every day for months. If you have drug-resistant TB, you're taking a lot more pills, and it can go up to 3 years of treatment. So it's very hard to get patients to adhere to treatment. And actually, in places where they have the resources, the way that you get people to take their TB drugs every day is you have to have a healthcare worker watch them take it every day. Yeah, so it's pretty intensive. And so one of the challenges, though, to TB is we actually don't know a lot about TB, the biology of it. We don't know how it's controlled by our immune system when it is controlled. We don't know why some people lose control of TB. Okay, so now I'm going to walk you through, this is my first really biology slide, and I'm going to tell you about the biology of this infection. So again, what happens, you breathe in TB, it goes into your lungs, and the cells that it goes into are cells called macrophages. Okay, this is really cool because macrophages are cells, their job is to ingest bacteria or other microbes that come in and kill, right? That's their job, that's what they're supposed to do. TB has evolved to make this cell its home. So TB lives inside the very cell that is supposed to be killing it. Okay, so early on after disease, the macrophages are completely defenseless. TB grows, so this line is telling you the amount of TB in the lung. So TB grows, unrestricted, probably for several weeks. Then what happens is this other cell type called a T cell comes in. T cells are able to activate macrophages to make them more powerful. T cells activate the macrophages, and then the macrophages are able to get some control of the infection, decrease bacterial numbers, but not eliminate the infection. So TB infection results in this stalemate. So this is what you were asking about, right? Latent disease. So in 90% of people, you get infected with TB. The immune system is somewhat effective. It's able to suppress the infection so that you don't have symptoms, but it's not able to eliminate it. And so 2 billion people probably have latent TB today. Most of them are going to be able to control the infection from their lifetimes. 10% of people will lose control of the infection. We don't really understand why. And the TB will grow again. Okay, so at every stage of this cell type that I've told you about, the macrophage, is really important. Okay, any questions about this before I keep going? Yeah?
Do you get infected with the primary disease when you already have latent disease? Does it just stay as latent, or do you get infected?
Yeah, that's a great question. So the question is, if you have latent disease, can you get infected again? The answer is yes, which is rare for infectious diseases, right? But it seems like the answer is yes. Whatever immune response that you have going on is not fully sufficient to protect you even from a reinfection. Yeah?
If you're immunosuppressed, so like for example a transplant patient,
Yeah.
and you've had latent TB, does it just become active then?
Yes.
Because of the immunosuppressants you're on?
Yes, so if you become immunosuppressed, If you have a transplant, and then that's actually a pretty severe immunosuppression, or even if you have an autoimmune disease and you take, let's say, Remicade or something like that, the first thing that your physician will do is test you for TB to make sure you don't have latent TB. If you test positive, you'll go on the drugs to try to eliminate it before they start the treatment. Because before they did that, there were a lot of people who would go on immunosuppressives and then, develop active disease. Yeah.
Can the macrophage reconsume the TB once it becomes active, or once it is active, does it just go straight into the disease itself?
So it just depends on the person. So sometimes the macrophage can kill and really suppress the bacteria, and sometimes it can't, and then you go straight to disease. Oh, I don't know why that happened. Okay. Yeah.
You get TB and it just becomes latent after that. It's just always there.
Yeah.
Everybody that gets it.
Yeah. I mean, maybe some people can eliminate it, but we know that so many people, the vast majority, don't. And the reason we know that is because people test positive, but they don't have disease. If they get something like HIV that immunosuppresses them, if they get something like if they go on a medication, then disease flares back. Yeah.
And so when it's latent, it's not communicated to others? Otherwise, I mean, if there are 2 billion people in the world that have this disease, you'd think that we would all be more likely to get it.
So latent people probably can transmit it much, much, much less frequently than people with active disease. Yeah, but if you'd asked me this 5 years ago, I would say latent people cannot transmit. But now there's data that actually people can transmit during latency. Yeah.
So how they get transmitted? Like if they're close to a person that has the disease, maybe?
Yeah, so if you have latent disease, you still have bacteria in there. And so some people with latent disease may have quite a lot of bacteria, it just hasn't risen to the level of symptoms. Yeah. Did I see a hand in the back of the room? Yeah.
When we have latent disease, when we catch a cold, how do we distinguish if it's a real cold versus reactivation?
Yeah. So if you have latent disease and you get a cold or something, how do you distinguish? The answer is that it can be challenging. And I'm not talking about this today, but I have a project in Uganda where we're trying to figure out how, in children actually, where it's particularly hard to do, how can you tell if someone has TB versus someone has a cold? So here, and many places, what you do is you get a chest x-ray. And so a cold doesn't look like TB on a chest x-ray. And then part of it is also the duration. A cold goes away. People usually, when they come into the clinic with TB, they've been coughing for months. Yeah. I mean, think about it. If you were coughing for 2 weeks, you probably wouldn't do anything. If it was a cold, it would probably go away. If you were coughing for a month or 6 weeks, then that's when you would start to think, oh, something might be going on, and you would go into the clinic. Yeah. Okay, great. Okay. So here are some important questions that we don't know the answers to. How do macrophages kill TB? Why do some people contain infection for a lifetime and others not? We know that the immune response can contain the infection. Are there ways that we could intervene to try to make it stronger, to eliminate the infection, or ensure that everyone who has the infection can contain it at the very minimum? And then also, how can we develop more drugs that can kill TB? Okay. So again, I'm hammering this point home, but the outcome of infection is determined by these macrophages. So if the macrophages can kill the infection is controlled. If the macrophages fail to kill the infection, then you'll have active TB. Okay. Now I'm going to take what is going to seem like a crazy detour, but we're going to come back around to this question about macrophages. So I'm showing you here a map that shows you the distribution of the most common mutation in humans. So this is mutation of a gene called ALDH2. And in yellow is what we would call the wild type allele of the ALDH2 gene. And in green is mutant. Okay? So if you have the mutant form, what it means is the protein doesn't work. So a gene encodes a protein, right? So, and the protein does the business of the cell. So if you have a mutant form, this protein doesn't work. And what you see here, I hope you can see it, is that there's a high degree of people in East and Southeast Asia who have this mutation. Okay. Has anyone heard of this mutation before? Yeah? Do you know what it causes? Aldehyde dehydrogenase? Yeah. Bingo. Okay. So when you've heard of this, it's because this ALDH2 protein is important for detoxifying ethanol. All right, so if you have a drink, ethanol is toxic. Your body wants to get rid of it. So how does it do that? The first step is this enzyme, alcohol dehydrogenase, converts ethanol into another molecule called acetaldehyde. Here I'm just depicting it with these stars. It's toxic, okay? So it has to be detoxified, and that's what ALDH2 does. ALDH2 converts this toxic aldehyde to a harmless compound. Okay, so what if you don't have a functional ALDH2? You drink a drink, and then you're stuck. You can't convert ethanol to you can't convert this toxic aldehyde to the harmless compound, so this toxic aldehyde builds up. This aldehyde, the reason it's toxic, it's very reactive. It can destroy proteins, DNA, RNA, it can harm cells. So when you drink, this aldehyde builds up in the blood, and it causes this characteristic flush, which I took this picture off of Wikipedia here, where you can see this person before a drink and after a drink. I probably didn't have to show this picture. Probably everyone knows someone who has this mutation, right? It's colloquially, it's known as Asian glow. So that's what we think of when we think of this mutation. But here's the thing. Why does this mutation exist? People who have this mutation have higher risk of cardiovascular disease and cancer. And so why would it be so highly prevalent in some parts of the world? Some people think it's because if you have this mutation, you drink ethanol much, much less, right? Because you feel sick when you drink. And so maybe that's the selective pressure. But there actually isn't very good evidence for that. And the reason that this mutation exists is still a mystery. Okay, so in my lab, one of the things that we study is we study the metabolism of macrophages and how the metabolism of macrophages changes when they get activated and they're trying to kill TB. And one of the things that we know is that actually these toxic molecules called aldehydes, they build up in these macrophages when the macrophages are trying to kill the TB. And some of these aldehydes that macrophages produce, so this is totally separate from ethanol. Macrophages are making these aldehydes too. It wasn't really understood why. But some of them are also detoxified by this enzyme ALDH2. So we developed this hypothesis that maybe what's happening is that if you don't have a copy of ALDH2 that works, when your macrophages get activated and they start making these aldehydes, they can't be detoxified. These toxic aldehydes build up. And actually, maybe they kill the bacteria and help you control infection. Does this make sense? Yeah.
How do you test this in the lab? Is it with animals or is it?
I'm glad you asked, because I'm going to tell you. Any other questions? OK. So how do we test this? OK, so the first thing is I don't normally do this, so I have to collaborate with people to look at humans. But how will we prove this hypothesis? First of all, is there any evidence that people who have this mutation have less TB? Second of all, this is just a hypothesis we have. Do these mutant macrophages actually make more of these toxic aldehydes? Even if they do they kill TB? Do they kill bacteria? And then finally, can we prove these things by infecting an animal and modeling this in the lab? OK, so science is really the most fun when you do it in a team and when you work with other people. So I just want to very briefly introduce the people who've worked on this project with me. This is my science best friend and collaborator, Heron Darwin at NYU. These are two awesome trainees in my lab, Scott, who's a graduate student, and Sam, who is a postdoc. And then this is Sarah Dunstan from Australia, and we collaborated with her because she actually studies TB in humans. And she has a cohort of patients that she follows in Vietnam and in Singapore. So when patients come into the clinic, they get diagnosed with TB, they actually sequence the genomes of these patients, and they sequence the genomes of the TB. So what we asked her to do is look at the sequence of the genomes of the people who are in this control group, so people who don't have TB, and then the people who have TB. And let's see if having the presence of the mutant ALDH2 changes your risk of having TB. Is this mutation enriched in the control group, which is what we would expect if it was protective against TB. Okay, and so what we found is that there's a 20% lower risk of having active pulmonary tuberculosis if you have this mutation. So in humans, it does seem like having this mutation protects you against TB. Okay. Yeah, no, great, this is great.
Am I reading that right, that your control group is from Singapore and your cohort was from Vietnam? Or is that
Oh no, that's a mistake. Thank you for pointing that out. Actually,
they're all from the same area?
these are all. This is just the TB numbers. I said it wrong. The control groups aren't on here. Yeah.
And both groups are from both countries, right?
Both groups are from both countries, so we're controlling within each country. Yeah. Good point. Excellent point. Are you a biology major? Were you one of the people that raised your hands? Yeah.
I'm manufacturing.
Oh, okay. Logic applies across disciplines. Okay, so cool. So does this prove that having this mutation somehow makes your body protective against TB? Yeah.
Does the mutation also mean that people who do get tuberculosis have less severe cases?
We did not have, it's a great question. We did not have the data to answer that question. Yeah, we only had yes, no, active TB or not. Yeah. Yeah?
So I know your research is all just about TB, but if you have that mutation, would it be against all bacteria? I mean, would they have a lower chance of getting most bacterial?
I can't believe these questions. You all are very sophisticated. Do you want to come work in my lab? I'm not talking about it today, but yes, they're protected against other bacterial infections that we've tested as well.
Not drink, but you're going to be sick.
Maybe. We don't have any epi data for other bacterial infections. And actually the ones, okay, this is kind of getting beyond what I was going to talk about today, but the ones where the mice that I'm going to tell you about seem most protected are opportunistic and kind of things that aren't normally pathogens but occasionally infect you. Yeah. Great questions. Okay. Wait, does this prove that there's something biological going on here, that these people are able to better control infection with TB? No. Because it could be that they just don't drink it, so they're healthier, right? We haven't really proven it yet. Good. Okay, so what we did is in the lab, we can take human macrophages and we can knock out ALDH2. Should have had a slide on this, but we also have mice that we've made in the lab that mimic the human ALDH2 mutation. Okay, so we can take these macrophages, and then first thing we asked is, do these macrophages actually make more aldehydes when we infect them with TB? Okay, so this is data from mouse macrophages, actually. So if we look at wild-type mouse macrophages, or we look at the ALDH2 knockout macrophages, here we're looking at one specific aldehyde, and what I hope you can appreciate is in the knockout, they're making a lot more of this aldehyde. How many chemists in the room? Any chemists in the room? Any people who work in mortuaries? Or because this aldehyde is formaldehyde. Our bodies make formaldehyde, actually, which blew my mind when I learned it. So our macrophages make formaldehyde when we get infected with TB, and ALDH2 knockout macrophages make more. Okay. So here I'm going to show you the infection with the mice. So we have mice where one group is wild type, one group is mutant for ALDH2. We infect them with TB, then we wait a month, and then we evaluate the bacterial load in the lung. So I want to note that this is a log scale, so each tick is a 10-fold difference. And when we compare at this time point the wild type versus the ALDH2 mutant, we see lower bacterial loads in the ALDH2 mutant. So what this is telling us is that this mutation does confer protection against TB that is independent of whatever lifestyle factors may be going on for the humans, right? So we have a lot more data than this, but in the interest of time, I'll just come back to this slide. So now we have evidence that humans with ALDH2 have less risk of TB. The macrophages from ALDH2 mutant individuals make more aldehydes. I didn't show you this data, but those aldehydes do kill TB and other bacteria. And we can show that mice that have this mutation and live in this carefully controlled environment in the lab actually have better control of infection. Okay, any questions? Yeah?
Do you have like any ongoing like investigations to answer any more questions?
Yeah, so one of the things that we're trying to do is I showed you that these macrophages make more aldehydes, but we don't actually fully understand the pathways that are leading to their generation. So we're trying to understand that. The other thing that we're doing is, when you look across population scales, okay, you don't need a very large change in your ability to control infection to affect kind of population genetics. But if you're a person, and I'm going to tell you, okay, you have this mutation, you're going to have 10-fold less bacteria in your lungs. That doesn't, you're like, okay, that's good, but that's not like awesome. You want the bacteria to be gone, right? So one of the things that it is awesome. I shouldn't say that. My results are awesome, but what you really want is eliminate the bacteria. So one of the things that we've found is that if you take the aldehydes that are generated and are higher in these mice, and you treat TB in combination with the antibiotics that we use for treating TB, you can actually get better killing faster. So now what we're looking at is, can we make a drug that will mimic this? And actually, we've discovered other pathways that also contribute. So can we make a drug that will make macrophages make lots of different aldehydes? And how does that affect kind of control of infection itself? And then how does that work together with antibiotics? Yeah.
So what does it take from here to get to a drug or a vaccine? How long is that?
What it takes is money and time. It's very, very challenging for TB. So when I was a postdoc, I actually worked directly on trying to discover new drugs. And I found a small molecule that is amazing in animals and looks just as good as the existing drugs. And it never went into clinical trials in people because it's really hard to find the funding for that. And it does happen and it can happen, but I would say on average to go from a discovery like this to a drug could take 10 to 20 years. A person's whole career, yeah.
So there are companies which are using RNA platforms to create vaccines for TB. Where do you stand?
In animal models, the mRNA vaccines for TB do not seem to be that effective. Yeah, unfortunately. A lot of people got excited after COVID. Oh, mRNA is going to be the magic bullet that's going to solve all the infectious disease problems, and I don't think that's the case. Yeah?
Do people ever get rid of COVID someday?
No.
You think it's permanent?
Yes. Yes. I think COVID is permanent. But we'll get to a point where, we already are getting to a point where it's much less virulent because we have immunity, and so it's not the same level of threat as it was when we were a completely naive population with no immunity. Yeah.
What are you, why are you working on this so much? Like, why is this your thing?
Why is TB my thing?
Yeah.
Because I'm a masochist? No, I'm just kidding. When I went to graduate school, I knew I wanted to work on immunology and infectious disease, and I knew I wanted to work on an infectious disease that was associated with poverty. I thought I was going to work on worms, hookworms. I went to UCSF, and I knew who I wanted to work with. And then I went and I heard a talk about TB, and I didn't even know TB was still a thing. I didn't know that it still was such a major problem, and I joined a lab on TB, and I completely fell in love with it. And I think it's so important, and I also think it's fascinating. So all of these things I told you about the kind of infectious cycle of TB, how it works, it's so different from other infections, right? And there's so much we don't understand about it. And so it's kind of also, it's an important problem and it's an endless puzzle. So that's why. Yeah.
If someone has the ALDH2 mutation and then they get TB, if they drink more while they already have TB, they not progress their TB?
Please don't drink to cure TB. No, it's a great question. And we actually have not done that. You know what? I get asked that almost every time I give a scientific seminar to scientists. I get asked that almost every time. And it's a great question. But the reason that we don't think that that will be the case is because alcohol is immunosuppressive. So alcohol drinking is a risk factor for TB because it really impairs your immune system's ability to control the infection. Yeah.
In an ideal world, like what would the best type of treatment be? Because I understand there's like the right line of drugs, there's like methamphetamine, but like
Oh yeah.
but I remember reading it in the Everything is Tuberculosis book that like it used to be $15,000 before like a lot of people protested, and it was reduced down to $1,500, and then malnutrition is also like a huge factor, and that's just
Yeah.
a problem in a lot of countries. So like in an ideal world.
You can't eradicate a disease without a vaccine. So in an ideal world, what we would have is a TB vaccine. And there have been, in recent years, there is one glimmer of hope that there is a vaccine that was tested in humans that reduced over 3 years, it reduced people's reactivation of disease by 50%. So that's really promising in the sense that it had some activity. We don't know. I mean, this has to work for 50 years, a vaccine, right? 3 years is a very short thing in the life cycle of TB. But it shows that there's some hope. The problem is we don't know why that vaccine worked. And then the other big vaccine trial that happened with TB didn't, and we don't know why. So that's something that I hope that people are trying to figure out. The Gates Foundation has the samples, and I know there's some process by which they're going to have them analyzed, but most of us don't know what that data is or even if they've done the analysis yet. Yeah?
What makes TB so uniquely difficult to make a vaccine for?
This is an excellent question. So what makes TB so difficult to make a vaccine for? Let me ask the audience a question. Do you know what is the mechanism of protection of the COVID-19 vaccine? What in your body is protecting you? Anybody know? Antibodies to spike, yes, antibodies. Antibodies are not protective against TB. So when vaccines work, for the most part, they work because they elicit neutralizing antibodies. TB actually is like, great, tag me with an antibody. That'll just help me get into a macrophage, which is where I want to be. So what we need for TB is we need a CD4 T cell response. We don't know how to engineer specific CD4 T cell responses with vaccines yet. And actually with TB, it's even harder because we don't even know what kind of CD4 T cells come in like a bunch of different flavors. We don't even know which flavor of CD4 vaccine that we would want for TB. Yeah?
From your graphic, it looked like the occurrence is much more in India and China and Southeast Asia. You talked about funding. Are you seeing funding come from that part of the world to help out this research?
India has a very robust internal research programs for TB. And in India, they're doing some of the most cutting edge science in the world on TB, actually, at some of the institutes there. In the past, in the US, it's hard to get NIH funding, but relatively speaking, there's a lot of it available. So in the past, we've never had to try to look to other countries for funding, and I don't know, that may be changing right now. But hopefully not. But India specifically, yes, has a very robust TB research. Yeah. Yeah.
Can I ask one more question?
Of course.
With the current administration cut back funding for everything under the sun, so to speak, how is that going to affect the research of this?
This is being live streamed, I'm just thinking. We're worried. So for TB research labs, there have been funding delays, and some of us have gotten stop work orders. That said, most of us, for the 2025 fiscal year, most of us ended up getting our funding. So for the people who got stop work orders, we worked with the NIH. They had some complaints about our research. We worked with the NIH to rebudget and try to change what we were doing to fit very arbitrary things that they were telling us about our research. But most people got their funding in 2025. The budgets that are right now in the House and the Senate, neither of them significantly slashes funding for the NIH. So as long as those pass, NIH funding should be stable for the next year. But there's a lot of things that happen internally at the NIH that determine how fast the funding comes or who gets it. And so I would say I'm worried, but I'm tentatively optimistic that things will be relatively normal next year. Yeah.
Test for TB, they do the thing in your arm.
Yes.
And then they do the chest X-ray. Are those the only two tests that there are?
No. So if they think that you have active TB, and I didn't mention this, but they will actually, they will try to get you to cough up sputum, and then your sputum will in adults has TB that can be hopefully detected. And then, so they'll take your sputum that you cough up, they'll put it in a machine, see if TB grows out. I always forget to mention that because I work, my human-related projects are with children, and children don't produce sputum, so we have a different set of challenges. Yeah.
How does the little TB test that some of us have gotten, how does that work?
How does that work? So what they do is, so these T cells that I was telling you about, when you have had TB, you will have T cells that recognize proteins from TB itself. And when you inject those proteins under the skin, the T cells that recognize those proteins will come and cause that swelling, that inflammation and swelling. So that's why it's specific, because they're putting in TB proteins, and then you get that inflammation as a result. Yeah.
And that's why you go, they ask you to do an X-ray in your arms after you have that inflammation, right? You have to make sure you don't have to do it.
Yes. I'm not a doctor. I have been a TB patient, but I'm not a doctor. So yes, in my case, in some of our cohorts in Uganda, they do. Most people in Uganda actually are PPD positive, and then here, if you're PPD positive, then they would give you a chest X-ray, and then a sputum test. Yeah.
If we did a similar test with different bacteria proteins, would they have the same effect if we had infections with other bacteria?
The thing about TB, I don't know, and it would be specific to the bacterium, because the thing about TB is that your T cell response lasts forever. And that's partially because you probably still have bacteria in there kind of continuing to stimulate them. So it would really depend on the specific bacteria. Yeah.
You said you were in drug development earlier in
Yeah.
your career. Have you seen any, I don't know if you still follow the drug
I do.
development world, but
Yeah.
have you seen any changes with the FDA's Orphan Drug Act and the GAINS program that incentivizes drugs to be developed in those cases?
Not specifically with those programs. But, okay, so I'm not going to tell you the second story that I was going to tell you, because it's better to get questions than to go through my slides. But, so this is the case, this is what, these are drugs that are used or have been used for treating TB. The first one was discovered in the 1940s. It's called streptomycin. It's no longer used. In the first clinical trial of streptomycin, resistance to streptomycin emerged. So that's one of the reasons why you give TB as a cocktail of drugs to try to prevent resistance. But here what you can see is all these TB drugs were discovered, and then there's this long gap, and then we have two, bedaquiline, which you know about if you've read Everything is Tuberculosis, or a million ways, I guess, and then protonamide. So we do have two new TB drugs, and this is really revolutionizing the treatment of drug-resistant tuberculosis. But resistance to these is already emerging, and so we do not have a robust pipeline for TB drugs. It's a big problem. I'll show you one more data slide. This is not my data, this is from the WHO, but drug-resistant TB is a huge problem worldwide. This is the percentage of new infections that are drug resistant here. This is 3% all the way up to more than 20%. So you can see that there are parts of the world where you're getting, here is, in South Africa, the drug resistance rates are, this is multi-drug resistance rates are like 5% or more. I mean, in Russia and the states of the former Soviet Union, it's more than 20% of drug resistant infections. So drug resistance is a huge problem, and we need more incentives for companies to work on TB, but there aren't very many that are interested in doing it. Okay, I saw, yeah.
So in the beginning of the lecture, you talked about like coughing up blood and how they would automatically know. Would that happen today?
Yeah, that still happens in places where people don't get, diagnosed and treated. Yeah, for sure.
Also, this is probably a stupid question.
Hmm?
Is TB like actually green?
Is it actually green? No, it's kind of a yellowy white. We grow it in the lab on plates so we can see it. And it's kind of yellowy white. We didn't talk about this today, but TB has a very thick outer wall. It's hard for antibiotics to get in. That's why we have to use specialized antibiotics. And that outer wall is made of kind of wax. And if you go into not anymore because we use forced air purifying protective thing, but when I was a graduate student and we just wore like an N95 mask in the lab with TB, you could smell it. It smelled like a candle. Okay. Did I see another hand over here? Oh, here, yeah.
I just had a question about the X-rays. What is it you're looking for? What's the marker on the X-ray?
There's some way that they can see inflammation, and I don't know how that, the radiologist would say this looks like TB versus something else. But one thing that is characteristic of TB is if you have advanced disease, you'll have what are called cavitations, and that's actually holes in the lung. So that's where you've lost a chunk of tissue out of your lung. Yeah.
Is the drug resistance, is that coming from mutating bacteria?
Yes.
Are there different variations of the bacteria?
So drug resistance comes when almost always in TB, drug resistance comes when the bacterium acquires a mutation in the target of the drug, so the drug can't bind anymore. That's the most common mechanism. There are different strains of TB around the world. One question that I didn't get asked, but which is a really good question, is if TB is so prevalent in sub-Saharan Africa, why is ALDH2 mutation not exist there? And we don't know the answer to that question, but one thing that we do know is that Asia has its own lineages of TB that are hypervirulent. They seem to be more virulent than other strains of TB isolated from different parts of the world. Yeah.
The way that this behaves and everything you explained, is TB kind of in a class? Is it like a one-of-one kind of thing, or are there other things that behave this way?
There are other diseases that have latent periods in the way that TB does, but in terms of driving mortality and its success as a pathogen, I think there's nothing like TB. The other big ones are malaria, but it has a very different life cycle, and it actually mostly, unfortunately, mostly the mortality is in children. As you develop immunity to malaria over your lifetime, you kind of become more naturally protected. And then there's HIV, and HIV is very different from TB because it's a zoonosis, which means it jumped, in the 20th century, it jumped from a non-human primate into humans, and so it's not as highly, it's very deadly, but it's not as highly adapted as TB is. TB's been evolving in humans for thousands of years. It probably was there, even before there was something we would call TB. There was probably something like that was in human, infecting our ancestors, our non-human ancestors, right? And so it's very uniquely evolved to do what it does. Yeah. Oh, let me answer someone who hasn't.
This might sound like a really stupid question, but the valley fever
Yeah.
that's what we're talking about, is there any kind of...
Yes, that's not a stupid question. That's a great question. Valley fever, very relevant to California. It's a fungal infection. It's often transmitted from the environment. So it's spores in the environment that you breathe in. But there are some similarities in kind of the symptoms and what happens when people get valley fever, as with TB. Yeah.
Is there any reason why like the post-Soviet states have such a high percentage of drug resistance?
Yes. There's a few reasons. One is at the breakdown of the Soviet Union, the public health infrastructure was significantly damaged. The main reason is probably that there are really high rates of incarceration and no real programs for preventing TB transmission in prisons. They also, for many years, did not have access to the drugs that are used for treating resistant TB. So if you got resistant TB, you couldn't be treated and you would just spread it. Yeah. Okay, one more question in the back.
Sorry. After being treated for TB, would you still test positive?
Yes. You will still test positive.
So it's always will be.
Yeah. I mean, some people it may wane in some people, but many people will remain positive by the skin test after that. Okay, this was so much fun. I'm happy to answer questions for a few minutes here at the front if you have other questions. Thank you for being an amazing audience. I can tell that many of you are Berkeley alum because you're awesome and smart and asking great questions. So enjoy the rest of your day and go Bears.